For more than a century, cancer has confronted medicine with a problem that has seemed almost impossible to solve. How do you teach the human immune system to recognize and destroy a disease that is not truly foreign, but rather a corrupted version of the body's own cells? A virus is an invader. A bacterium is an invader. Cancer is different. It begins inside us, using many of the same molecular building blocks as the healthy tissues surrounding it, which means that the immune system has to make an extraordinarily difficult distinction between what belongs in the body and what has become dangerous. Now, for the first time, scientists may have demonstrated in a large phase 3 clinical trial that a personalized mRNA treatment can give the immune system something it has never had before, a molecular blueprint tailored to the specific mutations of an individual patient's tumor. On August 19, 2026, Moderna and Merck announced that their personalized mRNA neoantigen therapy, known as Immunotherapy, or Immunotherapy, had met the primary endpoint of recurrence-free survival and a key secondary endpoint of distant metastasis-free survival. In the phase 3 INTerpath-01 trial involving patients with completely resected, high-risk melanoma, the trial compared the personalized treatment combined with Merck's checkpoint inhibitor Keytruda or pembrolizumab with Keytruda alone. The announcement matters for a reason that goes far beyond one company, one drug, or even one type of cancer. According to Merck and Moderna, this represents the first positive phase 3 readout for an individualized neoantigen therapy and the first positive phase 3 result for an mRNA-based cancer treatment. But before we call this a cancer cure, we need to understand exactly what has been achieved and what has not. This is not the kind of vaccine most people imagine when they hear the word vaccine. It is not a shot given to a healthy person that prevents cancer from ever appearing, in the way vaccines can prevent certain viral infections. Instead, this is an adjuvant treatment designed for patients whose melanoma has already been surgically removed, but who remain at substantial risk that microscopic cancer cells could still be hiding somewhere in the body. And that distinction is critical, because one of the most frustrating realities of modern oncology is that removing the visible tumor does not necessarily mean the cancer is gone. A surgeon may remove every tumor that can be seen on a scan or identified during an operation, and additional treatment may eliminate much of the remaining disease. Yet a tiny population of malignant cells can sometimes survive in places that cannot be detected. Those cells can remain dormant for months or even years before beginning to grow again, eventually producing a recurrence or spreading to distant organs. For decades, that possibility has represented one of the great unsolved problems in cancer treatment. What if, instead of simply trying to destroy every last cancer cell with drugs or radiation, we could teach the immune system to recognize the cancer so precisely that it could continue looking for those cells long after surgery was over? That is the idea behind the personalized cancer vaccine. The concept sounds almost deceptively simple. Identify the mutations that make a patient's tumor different from normal cells. Convert those mutations into an immunological blueprint. Deliver that blueprint using mRNA, and train the patient's own T cells to recognize the resulting tumor-specific signals. But making that idea work is extraordinarily complicated, because unlike a virus, cancer does not have one universal identity. Every tumor carries its own genetic history, and that is where the story becomes much more fascinating. The enemy within. Why cancer is so difficult for the immune system to recognize. Imagine that your immune system is a security force protecting an enormous city. Every day, millions of cells are inspected, damaged cells are removed, infections are detected, and potential threats are eliminated before they become dangerous. The system works because immune cells are constantly trying to distinguish what belongs to the body from what does not. When a virus enters the body, that distinction is relatively straightforward. Viral proteins are foreign, their molecular signatures are different from those produced by healthy human cells. And once the immune system learns to recognize those signatures, it can mount a targeted response. Cancer creates a far more difficult problem because the criminal was once part of the city. A cancer cell began as a normal human cell. It used the body's nutrients, followed the body's genetic instructions, and shared many of the same molecular characteristics as the healthy cells around it. Only after accumulating genetic alterations did it gradually acquire the ability to grow uncontrollably, evade normal biological restraints, invade surrounding tissue, and potentially spread throughout the body. The immune system therefore faces a dangerous dilemma. If it becomes too tolerant, cancer cells can hide in plain sight and continue growing. If it becomes too aggressive, it risks attacking healthy tissue. The challenge is not simply to make the immune system stronger. It is to make it more precise. For decades, scientists searched for a molecular feature that could distinguish a cancer cell from its healthy counterpart with enough accuracy to become a reliable immune target. Then genomics began to change the game. As sequencing technologies became faster and more powerful, researchers discovered that cancer cells carry enormous numbers of genetic alterations. Some mutations help drive cancer growth, while others are simply passengers that accumulate as the tumor evolves. But some of these mutations have a particularly important consequence. They can create proteins or fragments of proteins that are not normally found in healthy cells. When these abnormal proteins are processed inside the cancer cell, Small peptide fragments can be loaded onto HeLa molecules and displayed on the cell surface. And suddenly, the cancer cell is no longer completely invisible. It has left behind a molecular fingerprint. Scientists call these tumor-specific abnormal peptides neoantigens. Neoantigens are fascinating because they can provide the immune system with a way to distinguish a malignant cell from a normal cell carrying the same basic biological machinery. In effect, cancer may be forced to reveal a small part of its identity. The problem is that the fingerprint is different from patient to patient. A mutation found in one person's melanoma may not exist in another person's melanoma. Even two patients diagnosed with the same cancer type can have dramatically different tumor genomes, different HeLa types, different neoantigens, and different immune environments. This is one of the reasons cancer is so fundamentally different from an infectious disease. For a viral pathogen, millions of people can potentially receive the same vaccine because the target is shared. For a personalized cancer vaccine, the target may need to be discovered anew for every patient. And that changes the entire concept of pharmaceutical manufacturing. The traditional drug industry is built around standardization. Discover one molecule, manufacture millions of identical doses, and distribute them to large populations. Personalized cancer vaccination turns that model upside down. Instead of asking, what drug should we give this patient? The system begins by asking, what exactly is this patient's cancer? Only after answering that question can the medicine itself be designed. From tumor to blueprint, how do you design a vaccine for one person? The creation of a personalized cancer vaccine begins with something remarkably ordinary. A piece of the patient's tumor and a sample of their blood. The tumor contains the genetic alterations acquired by the cancer. The blood provides a reference for the patient's normal genetic background. By comparing the two, researchers can begin separating mutations that are specific to the tumor from the genetic information that belongs to the patient as a whole. Modern sequencing technologies can examine enormous amounts of genetic information, but the goal is not simply to produce the longest possible list of mutations. The real challenge is determining which mutations actually matter. A tumor may contain hundreds or even thousands of genetic alterations, but most will not make useful vaccine targets. Some mutations are not expressed at meaningful levels. Others may produce peptides that cannot be efficiently processed. Still, theirs may fail to bind the patient's HeLa molecules strongly enough to be displayed to T cells. And even if a peptide can be displayed, there is another question that may be even more important. Will the immune system actually recognize it? This is where computational biology and machine learning become increasingly important. The patient's HeLa genotype provides the immune system's molecular display framework, while the tumor's mutations provide a massive collection of potential targets. Computational algorithms can then rank candidate neoantigens according to multiple biological properties, including whether the mutated protein is likely to be processed into an appropriate peptide, whether that peptide is likely to bind the patient's HeLa molecules, whether the resulting complex can be presented on the cell surface, and how broadly the mutation is distributed across the tumor. That last point matters enormously. A target present in nearly every cancer cell may be far more valuable than one found in only a small subpopulation of the tumor. After this computational filtering process, researchers can select a much smaller set of high-priority neoantigens to encode into the personalized treatment. The goal is not to create a vaccine against melanoma in the abstract. The goal is to create a vaccine against this patient's melanoma. And that is the fundamental shift. Cancer treatment is moving from population-level targeting toward molecular individuality. Writing the immune system's instructions. Once the target neoantigens have been selected, scientists still have to solve another problem. How do you turn a list of mutations into a therapeutic message that human cells can actually read? This is where mRNA becomes so powerful. Messenger RNA is essentially an information molecule. It carries instructions that cells can use to produce proteins. Instead of manufacturing the final protein itself and injecting that protein into the patient, researchers can design an mRNA sequence that instructs the patient's own cells to produce the relevant antigenic material. In the personalized cancer vaccine approach, the selected neoantigens are incorporated into a carefully engineered mRNA construct. The sequence can be optimized for efficient expression, stability, and appropriate immune activation, while avoiding unnecessary immune sensing that could interfere with the desired response. The result is not simply a chemical compound. This is one of the most important conceptual differences between mRNA therapeutics and traditional drugs. A conventional drug is typically a finished molecule that acts directly on a biological target. mRNA is different. It carries instructions that allow cells to manufacture a biological product themselves. In the context of cancer vaccination, that means scientists can essentially program the immune system with a personalized molecular message. But there is still one enormous obstacle. mRNA is fragile. If it is injected into the body without protection, it can be rapidly degraded before it reaches the cells that need to read it. That is where lipid nanoparticles enter the story. The nanoscale delivery system. The mRNA needs protection, but protection alone is not enough. It also needs a delivery vehicle capable of getting it into cells. Lipid nanoparticles, or LNPs, provide that delivery system. The mRNA is formulated with carefully selected lipid components that self-assemble into nanoscale particles. These particles protect the RNA and facilitate its uptake by cells. Once inside the appropriate cells, the mRNA can be released and translated into the proteins encoded by the vaccine. In the immune system, antigen-presenting cells such as dendritic cells play a particularly important role. These cells are professional information processors. They collect molecular information from their environment, process it, and present antigenic fragments to T cells. The personalized mRNA vaccine essentially gives these cells a new set of instructions. Produce these tumor-associated targets. Process them, present them, and show the immune system what it needs to recognize. The dendritic cells then help activate tumor-specific T cells in the lymph nodes. This is where a personalized sequence designed on a computer becomes a biological response inside a living human being. And the transformation is remarkable. A tumor sample becomes genomic data. Genomic data becomes a list of mutations. Mutations become predicted neoantigens. Neoantigens become an mRNA sequence. The mRNA becomes a vaccine, and the vaccine becomes an immune response. The entire process is a biological information pipeline from patient to sequence, from sequence to molecule, and from molecule back to the patient's immune system. That is why personalized mRNA cancer therapy is more than simply another cancer drug. It is an attempt to turn genetic information itself into medicine. The immune army, teaching T cells what to hunt. Once the personalized vaccine is administered, the real biological battle begins. The vaccine does not directly seek out and destroy every cancer cell in the body. Instead, it helps the immune system learn what those cancer cells look like. Dendritic cells take up the vaccine, process the encoded information, and present the resulting antigens to T cells. Among the enormous diversity of T cells circulating through the human body, only certain populations will recognize the relevant antigen-HLA combinations. Those cells can then become activated and expand. Over time, the immune system can generate a larger population of tumor-reactive T cells capable of circulating through the blood and lymphatic system. The objective is to create a distributed surveillance network. If a residual melanoma cell remains somewhere in the body and displays one of the targeted neoantigens, a tumor-reactive T cell may be able to recognize it and initiate an attack. Cytotoxic T cells can form intimate contacts with target cells and release molecules such as perforin and granzymes that contribute to target cell destruction. But the most important idea is not simply that these cells can kill. It is that the immune system can potentially remember. Some activated T cells can differentiate into long-lived memory populations, providing a form of continuing surveillance that can persist long after the initial vaccination. That creates an intriguing possibility. Instead of trying to eliminate cancer using a treatment that disappears after the treatment course ends, medicine could potentially train the patient's immune system to continue searching for the disease. But there is a problem. Cancer has evolved specifically to survive inside the human body, and it has learned how to manipulate the immune system's own safety mechanisms. That brings us to the second half of this strategy. Why the vaccine needs a partner. The PD-1 problem. The immune system is powerful enough to destroy infected or abnormal cells, but that power comes with a serious danger. If T cells remain permanently activated, they can begin attacking healthy tissues. To prevent that, the immune system has evolved molecular checkpoints that function as brakes. One of the best known is the PD-1 pathway. Cancer cells can exploit this system. By expressing molecules such as PD-L1, tumors can engage inhibitory pathways on T cells and reduce their ability to attack. This is one reason checkpoint inhibitors such as pembrolizumab have transformed the treatment of several cancers. They interfere with inhibitory signaling and help restore T cell activity. But there is a fundamental limitation. Removing the brakes does not tell the immune system where to drive. If a tumor contains very few cancer-specific T cells, simply releasing inhibitory signals may not be enough. This is one of the central challenges in so-called cold tumors, where immune cell infiltration can be limited and the tumor microenvironment can actively suppress immune responses. This is where the personalized vaccine and the checkpoint inhibitor can complement one another. The vaccine is designed to generate and expand T cells that recognize specific tumor targets. The checkpoint inhibitor helps prevent the tumor from shutting those T cells down. One provides the target, the other helps preserve the attack. That creates a biologically intuitive combination. The vaccine teaches the immune system what to recognize, while checkpoint blockade helps prevent the tumor from putting the immune response back to sleep. This is not simply two cancer treatments placed next to each other. It is an attempt to solve two different problems in the same immune response. The first problem is recognition. The second is suppression. And if both problems can be addressed simultaneously, the immune system may have a much better chance of controlling residual disease. That is the scientific logic behind combining personalized neoantigen vaccination with PD-1 blockade. The real battle may be outside the body. The clinical breakthrough is extraordinary, but the next challenge may have very little to do with whether scientists can design the vaccine. It may come down to whether they can manufacture it quickly, consistently, and affordably enough for real-world medicine. Personalized therapy creates a manufacturing problem that conventional pharmaceuticals largely avoid. Every patient has a different tumor. Every tumor can produce a different set of neoantigens. Every personalized vaccine therefore begins with a different biological blueprint. That means the traditional pharmaceutical factory with enormous batches of identical products cannot simply be copied and scaled up. Instead, personalized cancer vaccination requires something closer to a highly flexible manufacturing network. The process begins with tumor acquisition and sequencing, followed by computational analysis and neoantigen selection. The individualized mRNA must then be manufactured, formulated, tested, released, transported under appropriate conditions, and finally administered to the patient. Every step takes time, and cancer patients do not have unlimited time. For some patients, the difference between 4 weeks and 8 weeks could be clinically meaningful. That is why the future of personalized cancer medicine will depend not only on better biology, but also on better engineering. Sequencing needs to become faster. Computational pipelines need to become more automated. Manufacturing systems need to become more flexible. Quality control needs to be integrated into the production process. And the entire workflow from tumor sample to finished treatment needs to become reliable enough that personalized medicine can operate at industrial scale. Cost is another challenge. Traditional medicines become cheaper when enormous numbers of identical doses can be manufactured. Personalized therapies cannot rely on exactly the same economies of scale. Instead, their economic advantage must come from automation, standardization of the underlying platform, and the ability to rapidly generate different products using the same manufacturing infrastructure. In other words, the future winner may not simply be the company that discovers the best neoantigen vaccine. It may be the company that figures out how to manufacture hundreds of thousands of different neoantigen vaccines without turning each one into a completely separate manufacturing project. That is a very different kind of pharmaceutical revolution. From cancer treatment to immune programming. And this is where the story becomes much bigger than melanoma. The most profound possibility of personalized cancer vaccination is not simply that one vaccine might reduce recurrence in one disease. It is that the same underlying platform could eventually be adapted to different tumors, different patients, and potentially different stages of disease. The traditional model of cancer treatment is largely reactive. A tumor appears. Doctors diagnose it. They determine its stage. They remove it, radiate it, attack it with drugs, or activate the immune system. But personalized immune programming suggests a different model. What if cancer could be monitored continuously at the molecular level? What if a blood test could detect evidence of residual disease before a tumor became visible on a scan? What if the genetic composition of the cancer could be tracked over time? And what if, when the tumor evolved, the treatment could be redesigned accordingly? This is where circulating tumor DNA or CTDNA becomes particularly interesting. A tumor can release small fragments of DNA into the bloodstream. As liquid biopsy technologies become more sensitive, researchers are exploring whether these molecular traces can provide an early warning of residual disease or recurrence. If that capability continues to improve, cancer surveillance could become increasingly molecular rather than purely anatomical. Instead of waiting until a tumor becomes large enough to appear on imaging, doctors could potentially detect changes in the tumor's genetic signature much earlier. And once those changes are detected, a personalized treatment could theoretically be updated. A new mutation could lead to a new target. A new target could lead to a new mRNA sequence. And a new sequence could become a new immune instruction. In that sense, personalized cancer medicine begins to resemble software engineering. The immune system receives information. The tumor evolves. The information is updated. The treatment is revised. The immune response is reinforced. Of course, biology is far more complicated than software, and cancer will not behave like a computer program that can simply be patched with a new version. But the analogy captures something important. Medicine is moving toward a world in which treatment may increasingly be generated from the patient's own biological data. What could the cancer clinic of the future look like? Imagine walking into an oncology center a decade from now after having a tumor surgically removed. Instead of simply waiting to see whether the cancer returns, your tumor and blood samples are immediately analyzed. The tumor is sequenced. Your normal DNA provides the reference. Your holotype is determined. Algorithms identify potential neoantigens. The most promising targets are selected. A personalized mRNA sequence is generated. And a manufacturing system produces your individualized vaccine. Treatment begins. But the process does not end there. You return to your normal life while your blood is periodically analyzed for molecular evidence of residual disease. If everything remains quiet, surveillance continues. If a molecular signal begins to emerge, the system investigates. If the tumor has evolved, the treatment strategy can potentially evolve with it. That is a radically different philosophy from the traditional model of cancer treatment. Instead of waiting for a visible recurrence and then launching another round of therapy, the goal would be to identify the disease at its earliest molecular stage and intervene before it has a chance to become clinically significant. The ultimate vision is not a single miracle injection. It is a closed-loop system of cancer detection, molecular analysis, immune programming, and continuous surveillance. And if that system can be made sufficiently accurate, fast, affordable, and clinically effective, it could fundamentally change the relationship between humans and cancer. Has cancer really been defeated? Now we need to step back from the excitement and ask the question that matters most. Has humanity finally defeated cancer? The scientifically responsible answer is no. Not yet. A successful phase 3 trial in high-risk melanoma is an extraordinary milestone, but melanoma is only one form of cancer, and cancer is not a single disease. Different tumors have different mutation patterns, different microenvironments, different mechanisms of immune suppression, and different abilities to evolve around treatment. The fact that a personalized mRNA approach has demonstrated benefit in resected melanoma does not mean the same strategy will automatically work against every tumor. Some cancers are relatively immunogenic. Others are extraordinarily difficult for the immune system to penetrate. Some tumors carry many mutations that can potentially generate neoantigens. Others carry far fewer obvious targets. And even when a T cell recognizes a cancer cell, the tumor can still attempt to escape. It may reduce antigen presentation. It may alter HLA expression. It may create an immunosuppressive microenvironment. It may recruit regulatory immune cells. It may produce factors that inhibit T cell function. Cancer is not a static enemy. It evolves, and that means the next generation of cancer immunotherapy will probably require combinations of strategies rather than one universal solution. There is also another question that cannot yet be answered by the latest phase 3 announcement. Will patients actually live longer? The trial has demonstrated benefit in recurrence-free survival and distant metastasis-free survival, which are critically important outcomes in the adjuvant treatment of melanoma. But long-term overall survival remains an essential part of the complete clinical picture. The scientific community will need to follow these patients over time. That is how medicine separates a promising result from a genuinely transformative therapy. The 3 technologies that changed the game. What makes this moment so interesting is that the breakthrough did not come from one technology appearing out of nowhere. It emerged because several technological revolutions finally converged. First came genomic sequencing, which gave scientists the ability to read cancer at the level of DNA. Then came computational biology and artificial intelligence, which increasingly gave scientists the ability to interpret enormous amounts of biological information and identify patterns that would be extremely difficult to find manually. And finally, advances in mRNA engineering and lipid nanoparticle delivery provided a way to turn biological information into a therapeutic instruction. Read the tumor, understand the tumor, program the immune system. That 3-step progression may ultimately prove to be more important than any single vaccine. For decades, cancer treatment often relied on the principle of overwhelming the disease with external force chemotherapy, radiation, targeted drugs, or combinations of these approaches. The new paradigm is different. Instead of asking only how to kill the cancer, scientists are increasingly asking how to give the immune system enough information to do the killing itself. That is a profound change in strategy. The beginning of a new era. So, has the cancer vaccine finally arrived? If by cancer vaccine you mean a universal injection that prevents every form of cancer, the answer is clearly no. But if you mean a new class of personalized therapies capable of taking the unique genetic fingerprint of an individual's tumor and converting that information into an immune response, then something genuinely historic may be happening. For the first time, the concept is no longer confined to laboratory experiments or early-stage clinical studies. A personalized mRNA neoantigen therapy has now produced a positive phase 3 result in high-risk melanoma when combined with pembrolizumab. The companies behind the program are continuing to study the approach across additional cancers, including non-small cell lung cancer, bladder cancer, and renal cell carcinoma. But the most important story is not Moderna. It is not Merck. And it is not even mRNA. The deeper story is that medicine is beginning to acquire something it has never truly possessed before, the ability to design a treatment around the individual genetic identity of a patient's disease. The tumor can be sequenced. Its mutations can be mapped. Its potential vulnerabilities can be predicted. And mRNA sequence can be designed. That information can be delivered into the body. And the immune system can be trained to recognize the target. If the cancer evolves, the strategy may evolve with it. If molecular monitoring becomes sensitive enough, recurrence may eventually be detected before it becomes visible. And if manufacturing becomes fast and inexpensive enough, personalized therapy could move from a technological marvel to a routine part of oncology. We are not at the end of the cancer story. We may be standing at the beginning of a completely different chapter. For generations, humanity fought cancer largely by trying to destroy it. Now, we are beginning to explore something more sophisticated, teaching the body to recognize it, remember it, monitor it, and respond to it. The ultimate goal is not to create one miracle drug for everyone. It is to create a system capable of turning the biological information inside each patient's tumor into a personalized defense. And that may be the most important idea behind the cancer vaccine revolution. Cancer is no longer simply something we hope to attack. It is becoming something we can increasingly learn to read, decode, and program against. The cancer vaccine may not be the end of the war, but it could be the moment when the rules of the war begin to change. And if personalized immune programming fulfills even a fraction of its promise, the question facing medicine may eventually shift from, how do we kill this cancer, to something far more powerful. How do we teach the patient's own immune system to find it and never stop looking? That is the promise of the mRNA cancer vaccine. And this revolution has only just begun.